So I'm Kenichi Maruyama, CEO at the GORYO Chemical from Japan. So I'd like to introduce the new intraoperative diagnostic system by using navigation drug. So navigation drug can reduce surgical margin through real time. So next slide shows information of navigation drug. The navigation drug can detect microcancerous tissue in real time during surgery. We deliver innovation by eliminating cancerous tissues in vivo using a 1-minute spray. So it has 3 characteristics. At first, navigation drug visualizes cancerous tissue with fluorescent light. The second, easy-to-use navigation drug achieve clean surgical margin without pathologist. The third, we enable high-quality cancer treatment without adding healthcare workload. So next slide shows the therapeutic rationale. The navigation drug can eliminate cancerous tissues by binding to a cancer-specific enzyme. So navigation drug consists of HMRG chemical structure. So HMRG has already proven its safety in Japanese clinical trials. As you can see, the HMRG structure. So HMRG has a 3 steps for detecting any cancers. First step, active ingredient GORYO binds to GGT high expressed in cancer cells. The second step, GGT catalyzes GORYO to produce fluorescent HMRG. The last, lysosome quickly absorbs HMRG, enabling localization to the cancer cell. Okay, next slide. So if you use navigation drug to breast cancer, navigation drug improve treatment without adding healthcare workload. Broadening ability to non-pathologists, the navigation drug lowers recurrence and reacquisition of breast cancer. As you can see, background in United States, so annual diagnosis rate is 180,000 per year, and high recurrence caused by untreated surgical margins. If you use navigation drug, we provide 3 benefits. One benefit is availability, increased use for non-pathologists. The second, savings in labor, diagnosis without pathologist. The third, reduced reacquisition by more complete cancer removal. Okay, next slide shows market potential. Actually, we have already investigated the market potential in the United States. As a result, close to 65% of surgeons interested in using the navigation drug in lumpectomy. So this data is based on a survey of 75 surgeons in the United States. As you can see, DCIS is shown around 6%. Oh, DCIS means the ductal carcinoma in situ. And both the invasive and the weighted are shown around the 65% respectively. So, okay, next slide shows how to use the navigation drug. It is very simple. 5 easy steps within 30 minutes and without pathologist. The navigation drug follows simple steps and it's easy to operate. At first, remove tumor from the breast, and then prepare the sample. It is no special technique required. The third step, treat a navigation drug by the drop or spray. And after 20 minutes, you can see the signal if you have the tumor. And then, if you see fluorescent signal, it means positive, or not means negative. It's very simple. Okay, next slide shows the clinical study result in Japan. So we have already got the high efficacy regarding the naloxone drug to breast cancer. So the sensitivity is around 80%, and the selectivity is 91% selectivity. This data based on the fluorescence response of 274 patients. As you can see, distribution of fluorescence response. So in this data, so above the threshold means malignant, and under the threshold means benign. So as you can see, the threshold clearly divides from malignant and benign. Okay, so next slide shows the growth potential. Naloxone drug provide scalable solution. So other tumors and diseases, various diseases could be treated with the naloxone drug as is or with minor modification. So after developing breast cancer, we have two strategies. One strategy is diseases treated with amino acid modification. So this means that if you change X part of HMRG, you can detect from esophageal cancer, colorectal cancer, gastric cancer, and others. So next strategy is diseases treatable with existing substrate. Existing substrate means the dipeptide. Dipeptide is the LM. L means leucine, M means methionine. So LMHRG can detect from glaucoma, pancreatic leak, and brain tumor, and lung cancer by using just one LMHRG. Okay, so this is the last slide. So this slide shows the regulatory update in Japan. We have the 5 pipelines. One pipeline is in vitro diagnostics, like breast cancer. And the others is in vivo diagnostics. So actually, we have already out-licensed breast cancer to Thermo company in Japan. And in vivo diagnosis, the glaucoma also have been out-licensed to Senju Pharmaceutical in Japan. So in this conference, we are seeking a collaborative partner which can develop breast cancer in United States as well as Japan. Okay, so that's all for my presentation. Thank you very much. Thank you for your attention.